Hello, I'm DNA. You may be wondering what I have to do with RNA. Well, according to the central dogma, all RNA has to come from me. In a long process called transcription, RNA is produced from small segments of myself. This is done using the enzyme RNA polymerase. However, RNA polymerase can't do everything. What it first needs in order to begin its process of transcription is a promoter. In the first stage of transcription, called initiation, RNA polymerase finds the promoter and binds to it. Once RNA polymerase is bound to the promoter, it begins to separate me apart into two individual strands. The next part of the transcription process is called elongation. Once RNA polymerase is bound to the promoter and began to separate the two strands of me, it begins to replicate the DNA based on the template strand. It does so beginning at the 3' end and ending in the 5' end. However, RNA polymerase is a workhorse, so it will continue to replicate the DNA from the template strand going in the 3' to 5' end until it reaches something we call the terminator. Once RNA polymerase has reached the terminator, it can detach. What's left is the RNA. In bacterial cells, the RNA produced can immediately be used as mRNA or other forms of RNA. However, in eukaryotic cells, the RNA produced has to be processed.
Now moving on to how does DNA even relate to RNA? So the enzyme that converts DNA into mRNA is called RNA polymerase, which attaches to the DNA double helix. The RNA polymerase can unwind the double helix and begin to copy one of the strands of the DNA to form an mRNA transcription of the gene. So the gene in the DNA encodes protein molecules of the cell. The first step will involve DNA, where DNA will transfer information to a messenger RNA molecule. This process is called transcription. Now, transcription within DNA of a gene will become a template for the complementary base pairing and the enzyme called RNA polymerase. And during transcription, the gene expression will allow for mRNA to read according to the genetic code. This will relate to the amino acid sequence in proteins. Each group of the 3 base pairs in mRNA will be considered a codon. And each codon specifies an amino acid. Therefore, the mRNA sequence in the template to assemble in a chain of amino acids that will form a protein. The amino acid is specified by each mRNA codon, and multiple codons can code for the same amino acid. So the DNA can relate to, more specifically, mRNA in order to create proteins, and that is how DNA actually will aid in transcription for mRNA.
These base codons will then go to its proper amino acid in order to create a protein chain. mRNA is responsible for creating the protein chain needed in the replication process. Now for our next speaker. And then carries that information to the cytoplasm, where cytoplasm creates protein. But how exactly is protein created in cytoplasm? From the information gathered from the DNA. Well, in the cytoplasm, the mRNA sequence is read and then will be translated into 3 base codons. These base codons will then go to its proper amino acid in order to create a protein chain. mRNA is responsible for creating the protein chain needed in the replication process. Now for our next speaker.
Hi everyone, so I'm Nice Turn and I'll be acting as the tRNA. And basically what I am is a small RNA molecule that basically it plays a big role in the protein synthetic. I am basically like a little Uber, which is like a link and an adapter between this amino acid and this peptide chain and the ribosome. And basically what I do is I carry this amino acid, and what I do is I take it along down that little path which will go to the ribosome. And basically what I do is this amino acid, the mRNA will take this with the amino acid gone through, and it will basically let out a peptide chain with the 3 nucleotides going on. And which during that time, the mRNA will tell me, the tRNA that's carrying the amino acid, I will then connect the amino acids with the 3 nucleotides into one of the peptide chains. And which is why I am a T-shaped. And the top part of me is basically the anticodon that's holding this amino acid that I'm carrying. And that's what I am.
Hi, my name is Abby, and I will be talking about rRNA. So the R in RNA stands for ribosomal, and we know that that stems from ribosomes. Ribosomes are an important part of protein synthesis. So in the central dogma, the rRNA is a huge contributing factor to making protein. RNA contributes to the overall form of a ribosome, and that is because one rRNA, with this, let's say that this is one RNA strand, and a lot of protein molecules together, they form one small subunit. And a ribosome is made of two subunits, one small and one large. And the large subunit is made of 3 strands of RNA plus a lot of protein molecules. So let's call this the small subunit. This would be a small subunit, this would be a large subunit. Together, they would constitute a ribosome, and they have binding signals for tRNA. The entire ribosome will be involved with translating an mRNA strand into protein. So this would be so now we're changing it up and using this as a ribosome. This bottom here would be the small subunit, and this would be the large subunit, but together they make a ribosome, and this is whole. And this would be the mRNA strand. And together, it would pretty much just read the strand and the genetic code that's in here in order to find out how exactly to make a protein. And it'll find signals with the stop codon and create a protein through here. And now let's move on to the next person.
And to further conclude everything, we wanted to ask and answer, why does this information even matter? Well, by learning about DNA and RNA, we can actually understand how the human body works. By further understanding this information, we can do a lot more research and help others in that process.